Hello, everyone. It's my pleasure today to welcome you all to this webinar entitled Advancing Targeted Protein Degradation Research with Automated Western Blotting. My name is Hannah Maple. I'm the Innovation Manager for Tocris, a Bio-Techne brand, and I will be moderating the webinar today on behalf of Bio-Techne, Aurelia Bioscience, and Labroots. This webinar will be approximately 1 hour long, and the agenda today is as follows. I will provide a short introduction to targeted protein degradation. My colleague, Carsten, will then provide an explanation of the simple Western technology that underpins the work that will be presented today. And then I will be inviting Gary Allenby from Aurelia Bioscience to present today's research talk. We will be ending with a live panel Q&A, so please do submit your questions to us using the Ask a Question box throughout the session. We'll answer as many questions as we have time for at the end of the presentation. I'd like to take this opportunity to introduce today's speakers and panelists. Dr. Gary Allenby will be giving today's research presentation. Gary is the founder and chief executive officer of Aurelia Bioscience, who are a UK-based preclinical contract research organisation specialising in bioassays and screening. Gary gained his PhD from Edinburgh University before moving into the pharmaceutical industry with positions at Hoffmann La Roche, GlaxoSmithKline, and AstraZeneca. He brings a wealth of experience in assay development and implementation. On the panel for today's Q&A and providing introductory information on the products that supported Gary's work are myself, Hannah Maple, and my colleague, Carsten Luke. I manage the targeted protein degradation range at Tocris, and I'm working on development and commercialization of tools and technologies to support this growing research field. Dr. Carsten Luke is the Field Application Science and Science Manager for ProteinSimple. Carsten has a PhD in proteomics from the TU Munich and has extensive experience in electrophoresis and other protein separation methods. Carsten is highly experienced in the Simple Western platform for automated Western blotting and will be able to provide a detailed introduction to this technology. I'd like to open this webinar with a brief introduction to targeted protein degradation and PROTACs. This is a relatively new research field that harnesses the cell's own ubiquitin proteasome system to selectively degrade or knock down target proteins within cells. It is a tremendously powerful approach with significant potential, both as a research tool modality and for clinical development. A PROTAC, also known as a degrader, is a hetero-bifunctional small molecule that contains two ligands joined by a chemical linker. This is illustrated schematically in this diagram. One ligand is designed to bind to an E3 ligase enzyme, shown here on the left, and the other ligand is specific for a target protein, shown on the right in green. By simultaneously binding these two proteins, the PROTAC degrader induces a ternary complex, which can initiate transfer of ubiquitin onto the surface lysine residue of the target protein. The cell's proteasome recognizes the polyubiquitinated target protein as marked for destruction and degrades it into small peptide fragments. The degrader, or PROTAC itself, is not destroyed and can go on to induce ubiquitination of further target protein molecules. The process is therefore classed as catalytic, which means that only a very small dose of degrader is required to elicit a strong and sustained response. We are building our portfolio to support researchers working in this field from a number of different angles, as illustrated on this slide. For researchers investigating E3 ligase biology or screening for new E3 ligase ligands, we have the leading range of E3 ligase and related proteins. For researchers wanting to test degrader technology or develop their own degraders, we have a suite of chemical tools available. For target validation in this space, we have commercialized the TAG degradation platform. And finally, for all degrader development workflows, appropriate assays are critical and will be the subject of today's talk. Bringing all of this together, this slide presents Bio-Techne's complete solution for targeted protein degradation research and the flagship product lines that support each aspect of this workflow. I'm going to finish here by providing you with the link to our targeted protein degradation homepage, www.tocris.com/TPD, where you'll be able to find more information on all of the workflow and each product line, as well as supporting literature, animations and videos explaining the technology in more detail, and also our new PROTAC panel builder tool for requesting bespoke panels of chemical building blocks for your degrader discovery programs. With this, I would like to pass you on to my colleague, Dr. Carsten Luck, who will be providing an overview of the automated Western blotting technology used for today's research talk. Over to you, Carsten.
Thanks a lot, Hannah, for the nice introduction. As introduced by Hannah, my name is Dr. Carsten Luck. I'm the Affairs and Science Manager for EMEA. I'm with ProteinSimple Bio-Techne for more than 10 years. And first, I would like again to thank you for attending this webinar. And today it is my pleasure to introduce you to the Simple Western technology, the new gold standard in Western blotting. Proteins are our core, and we share this with the R&D system and the Novus brand. Both R&D system and Novus provide a wide variety of proteins and antibodies to help support your research. ProteinSimple provides the analytical tools designed to make your protein analysis simpler. What you see here might be really familiar to you. This is the pain you feel and must live day by day whilst using the Western blot technology. On top left, you see an inverted smile effect. Below, you see bubbles in your transfer arm. Anyhow, the transfer, you got an unknown transfer efficiency. The picture on the right, you see this is the gear you need, a lot of gear. And at the end, using the Western blot technology, it is semi-quantitative at its best. And as you know, the Western blot technology requires a lot of manual steps. Many steps mean many sources for issues and errors, meaning many things which can go wrong. And it takes a long time, and I repeat myself here, it is at its best semi-quantitative. And my question to you now is, do you know when the Western blot was invented? It was 1979. It's still used in the same way. Yes, it is still a source which you can use to gain good results, and it's still used heavily in a lot of labs. But let me ask you one question. Are you still using your Walkman? The Walkman was invented as well in 1979. And I personally, I don't know so many people which are still using their Walkman. So they nowadays use a mobile phone where they stream the music or have their MP3 files on it. So I would like you to welcome to the 21st century. Welcome to the Simple Western technology. So our technology is easy to use and gives you quantitative results in 3 hours. Here you prepare your samples literally the same way as for a traditional Western blot. You dilute your samples and you denature them by heat. The main difference is that you don't load your samples on a SDS gel and you blot them afterwards. Here you load 3 microliters of your sample along with the primary antibody and secondary antibody in our plate. For secondary antibodies, you can either choose the ones for NIR or IR fluorescent detection, or a HRP-coupled ones for chemiluminescence detection. The plate itself is pre-filled with the separation reagents. If you choose chemiluminescence for detection, you need to add as well luminol and peroxide. After plate preparation, you spin the plate to get rid of bubbles. Next, you peel off the protection foil for the separation reagents and you place the plate into the instrument. Next step is adding a capillary cartridge with 25 empty capillaries into the Jess instrument. You close the door and you start the run. After 3 hours, you get your data with high precision and reproducibility. But what happens? What happens inside the instrument? What happens inside the capillary? So the Jess instrument separates your proteins by size. This means our empty capillaries are first filled with matrices, a separation and a stacking matrix. If you look at the top left corner, the filled capillary, if you would rotate this capillary 90 degrees clockwise, you would have the same pattern as a normal SDS gel, stacking and separation. In a second step, we load a small amount of sample into each capillary. After this step, we apply electricity, current flow, and the negatively charged proteins move to the right side, the anode, anodic side, plus side. Whilst running through the matrix, they get separated by size. Small molecules run faster than large ones. This is not new. This is how capillary electrophoresis has worked for decades. The new added part comes now. Our caps have a special proprietary coating on the inside of the capillary. By using UV light, this coating is activated and it binds all proteins next to the wall. In the following wash step, we remove everything except the covalently bound proteins to the wall. The empty space inside the capillary is used to apply washing steps and the incubation steps for the immune detection. Having a glass capillary, we can either detect the light signal from a chemiluminescence reaction, or we can excite the fluorescent secondaries and measure their emission inside the capillary. At the end, we record the images in the results in a series of images over time with a built-in camera. The following animation should describe those steps in a more animated way.
Simple Western immunoassays take place in a capillary. Your sample, separation matrix, stacking matrix, antibodies, and reagents are loaded automatically from a specially designed plate. Jess begins by aspirating the separation matrix and then the stacking matrix into each capillary. Next, your protein lysate is loaded, and capillaries are lowered to make contact with running buffer. Voltage is applied to enable separation by molecular weight. Once the separation is complete, UV light immobilizes the proteins to the capillary wall. With proteins now immobilized and the matrix cleared of the capillary, Jess starts the immunoprobing process. For chemiluminescent detection, samples are first incubated with the primary antibody, followed by a secondary HRP conjugate, and finally chemiluminescent substrate. The chemiluminescent reaction is recorded by a CCD camera in a series of images over time. For fluorescent detection, samples are incubated with the primary antibody, followed by infrared or near-infrared fluorescent secondary tagged antibodies. Excitation of the fluorophores releases photons, and the emission spectra is detected and recorded by a CCD camera in a series of images over time. Protein normalization has never been easier with Jess. Just load her protein normalization reagent into a row of wells on the plate, and she'll take care of the rest. The fluorescent labeled reagent reacts with amines on proteins, enabling a between capillary comparison of protein load. Best of all, Jess's fluorescent detection capabilities enable two-color protein detection for multiplexing on top of protein normalization.
So with our technology, you get Western blot-like results with a differentiator that in our technology, you have much faster time to result. In our platform, we are capable to do real quantitation. You get a high reproducibility. We have the possibility to multiplex, meaning detecting different targets from the same sample in the same capillary. And you only need a very low amount of sample. As you could see in the animation, we do offer as well an in-capillary protein normalization using a fluorescence probe, taking off the burden from your shoulders to test and run several loading controls. At the end of July, we will launch this, what we call the replex feature, which you can use as in this case as well, to detect low abundant targets or low abundant proteins with a more sensitive protein labeling reagent, which is then as well used in the same capillary. So even you could say that we are the new kids on the block. Our technology is used to publish frequently. And as you can see on the left side, the uptake on our technology is going up, you could say, day by day nearly. And the way people do publish is pretty similar to the way you would publish standard traditional Western blot data. It's not a problem. And as you can see on the example I show here, you can even publish it together with the data you already received before having our technology by hand. So I hope I could give you, with this short introduction, a view on our technology and the benefits which come with it. And now it's my real pleasure to hand over to Gary, who is showing you what is possible with our instrument in a degrader research.
Fantastic. Thank you, Carsten. Thank you, Hannah. My name is Gary Allenby. I'm a member of Aurelia Bioscience. What I'd like to talk to you about today is the utilization of the technology that Carsten's just been talking about, and also some of the compounds that are available from Tocris in order to look at PROTAC in different backgrounds. So I think really Carsten's covered this slide. So I was just going to describe the technology, but he has done it so well that I'll just move on to the next slide. So safe as to say that the reason I put this slide in was I wanted to show the two readouts that the instrument can give you. So on the right-hand side, you can see what anybody who's seen Western blots in scientific publications would say is a typical appearance of a Western blot. And really, that's a pseudo image that the instrument produces on the screen following a run, and thereby gives you an idea of the molecular weight of your species that you're looking at, and also the intensity of that. However, on the left-hand side, you can see a graph, which on the x-axis is molecular weight, and on the y-axis is chemiluminescence. And what this gives you is an indication of the amount of protein, judged by the chemiluminescence, that you have in that lane. Yeah. So we originally purchased a WES, which gave us the opportunity to just do chemiluminescence about 4 or 5 years ago. Utilized that instrument quite significantly, and then more recently, at the beginning of the year, we bought a JES. The difference being that JES is the sister of WES, and JES really gives you the opportunity to do both chemiluminescence and fluorescence. So by utilizing a secondary antibody that's labeled with a near-infrared dye or an infrared dye, you're able then to both quantitate and multiplex two species or more species in the same lane. So just to highlight, because Hannah's done a really good job with regard to PROTAC, but just to highlight, we see this as a phenomenal approach in drug discovery. with the ability to be able to deplete protein specifically from within cell types, simply by utilizing the endogenous machinery, i.e. the E3 ligase and ubiquitination within the cell. And so what that gives you the opportunity to do is to massively deplete, if not knock out altogether, the protein of interest by utilizing what is a relatively small molecule. And the reason I say relatively is because often these molecules can be in the 700 to 1,000 molecular weight range. So in addition to that, if we think about dissecting the design of a degrader molecule, it's really composed of three components. The ligase binding section, shown in blue on the left, which obviously binds to the ligase and brings the protein of interest in close proximity with the ligase by utilizing the molecule which is shown in the green on the right-hand side. So that molecule, you would hope, specifically binds to your protein of interest. Now, the interesting thing in terms of drug discovery is the fact that your protein of interest doesn't necessarily need to be an enzyme. So you don't need to be binding the binding site to the enzyme. You could actually utilize a binding site which is on the protein itself, and just by using that, drag the protein to the ligase. And then in the center is what people call a linker domain. So a flexible or statically hindered section of molecule that really allows the movement of both the ligase and the target warhead in order to bind both species and to allow ubiquitination to occur. So my presentation summary, we're going to discuss really two targets, CDK9 and CDK6. And we obtained two molecules from the Tokras catalog, which are the THAL SNS032 and BSJ03123, which I'll just refer to as CK6 and 9 degraders. Obviously, CDK9 is involved in transcription, and CDK6 is involved in the regulation of the cell cycle. And so both of these kinases are particularly important in cancer and in cancer treatment. So what we're going to show in this presentation are dose responses from PROTAC molecules, the ones that I've mentioned, the impact of incubation time with the PROTAC molecule on the cells, and what that looks like in terms of protein degradation, and then choosing the right cell background. So think about the therapy area, but also think about the type of cell line that you want to utilize to try and exemplar your PROTAC. And then really investigating PROTAC activity in the presence of cyclohexamide, which obviously would block protein production, and also in terms of a proteasome inhibitor, which is then going to prevent the degradation. Despite the ubiquitination, it would prevent the degradation of your protein of target. And then lastly, we'll go through some of the pitfalls, some of the hurdles, and some of the things that we've encountered whilst we've been doing this work. We've really been doing PROTAC work over the last 12 to 18 months, I would say, and gained experience as a consequence. So CDK9 to look at initially. So many of my slides will have the pseudo blot on the right-hand side, and we'll have a graph on the left-hand side. Each one of the lanes, as I would call them, as you can see at the top, is labeled with the treatment. So the first lane there is DMSO, 4 hours, 0.3 nanomolar, 4 hours of the CK9, PROTAC, etc., going from left to right. So we go from low concentration to high concentration, from left to right. CDK9 is in the chemi channel. So by that, what I mean is that we're using the luminol and the peroxide in order to generate a chemiluminescent signal. And what we've done is in each of the lanes, we've multiplexed that with actin. So you can see at the bottom, the red bar is actually the quantity of actin which is in each one of these lanes. And so we've multiplexed the two in the one lane. Now we utilize the JEST to do this because it can do both chemiluminescence and also fluorescence. So we treated these cells Now, these cells are MV411 cells, and they're a beta myeloid monocytic leukemia cell line, so they're a suspension cell line. And we treated those with low to high concentrations of this CDK9 PROTAC. And as you can see, what happens is that we can see two bands in the in lanes, but you can see that the lower band is depleted and then is completely removed by concentrations in the range of 30 nanomolar up to 300 nanomolar in just 4 hours. So I really found this really startling and quite exciting, the fact that you can completely deplete a cell, in this case of CDK9, within 4 hours of treatment with the compound at relatively low concentrations. So if you look at the next slide, similar sort of layout to the slide, but in this case we're using the CDK6 PROTAC. And what you can see is that we get about 70% of the CDK6 knocked down at 30 nanomolar above after 4 hours. Yeah. So in this case we do have two bands, a CDK6 upper band and a CDK6 lower band at around about 40 kilodaltons. And as you can see, it is depleted. It doesn't completely disappear. It gets knocked down to approximately 0.25. So what we do, I should have mentioned in the last slide, but what we do is that we normalize our data to the actin band. And the reason for doing that is obviously because we want to normalize in terms of the amount of protein and in terms of the housekeeping protein, in this case that we use in actin. Every cell lysate that we generate, we do a BCA assay on it in order to determine what the protein concentration is. And then by knowing that, we balance our protein concentrations, but we utilize the actin in this case in order to get an exact ratio of the amount of CDK6 to the amount of actin. And so as I say, you can see here that we deplete by 70% with this PROTAC after 4 hours in this particular cell line. Fascinatingly, and I never thought you could do this until my colleague Rachel showed me this, is the fact that you can actually draw a graph which actually looks like a pretty nice dose response curve in spite of the fact that you're using Western blot data. So what you can see on the left-hand side is normalized CDK9 to actin against the concentration of the CDK9 PROTAC, and on the right-hand side, the CDK6 PROTAC. So this is a DC50 plot, and you can see that we're in the range of 11 nanomolar for CDK9 and 6 nanomolar for CDK6 for the removal of those two particular proteins. Really interesting from the point of view of then trying to rank compounds, rank PROTACs against each other based on Western blotting. In this case, we then went on to look at the time dependency of the activity of these compounds. In this case, I'm showing CDK9, and we're comparing the data that we obtained from 4 hours versus the data that we obtained for 24 hours in the same cell type, these MV411 cells. And you can see that the DC50 is shifted slightly to the left with increasing time, and basically we've increased the potency of our compound from 11 nanomolar to 2 nanomolar. We do get a little bit of fluctuation. So each one of these points is N of 3. So basically 3 lanes run with the cell lysate. And you can see that both the PROTAC activity can fluctuate with time, but also we seem to see a bit of a hook effect at the lower concentration. So that was CDK9. So what does what does CDK9 look like if we compare and contrast between two different cell types? So in this case, what we're doing here is we're looking at a background of either HEK293 cells or MV411 cells. Now, obviously, they're completely different cell types. One is adherent, one is suspension. So what you can see with regard to CDK9 is that this PROTAC also works in HEK cells, in addition to working in MV411s, but it's actually less potent in HEK cells in comparison with the MV411s. But again, you can see we've got actin in the near infrared channel, which is the red bars, and you can also see the chemiluminescence on the CDK9 in the black bars. So you still get activity with this PROTAC for CDK9 in these two different cell types. If, however, we look at CDK6 using the CDK6 PROTAC molecule and compare and contrast between the two different cell types, what you'll be able to see is that whilst we can see degradation of CDK6 in the MV411s, we don't see any degradation of CDK6 in the HEK293 cells, yeah, which seems to suggest that either the compound doesn't particularly have an effect after 4 hours in the HEK293 cell line, and that makes you consider what cell type background you should be working in when it comes to looking at the activity of your PROTAC molecules. And I should also say that in every case where I present the data in this way, both the near infrared actin and the CDK6 are multiplexed and measured in the same lane. Okay, so to then move on and look at some mechanistic data. So we know that there's a compound called epoxamycin, which is naturally occurring and is selective for inhibition of the proteasome. And so what that was resulting is in a blockade of the ability of the cell to be able to degrade the target protein, irrespective of the presence or absence of the PROTAC molecule. And also, we could obviously use something like cyclohexamide, which is a protein synthesis inhibitor, in order to prevent the generation of new protein. And now, interestingly, in terms of biology, cells make proteins for specific reasons, and generally there will be some sort of feedback loop, which will then switch off the protein production if the specific protein has done its job in the cell. So in many cases, what you'll see is that when we show plots or graphs, you'll see that sometimes it doesn't go to zero, i.e. maximum inhibition and a total lack of protein in the cell. And probably one of the reasons for that is because we're degrading the protein that is present in the cell over the course of the incubation time, but at the same time, the cell is making more protein because that protein is not in high enough concentration to actually do its job and to switch off its own manufacture. And so here what we've done is that we've added the epoxamycin for 4 hours in the presence of the CDK9 PROTAC in the MV411 cells. Yeah, and on the right-hand side, you can just see the labels of each one of the lanes. So the first 3 lanes are DMSO, the second 3 lanes are DMSO plus 1 micromolar epoxamycin. The third 3 lanes are our CDK9 compound at a concentration that would completely degrade or aid the degradation of CDK9. And in the last 3 lanes, it's the same concentration of the PROTAC, but in this case, we've also added 1 micromolar epoxamycin, right? And so what you can see is that we get approximately with 300 nanomolar CDK9 alone, we get a 90% reduction in the CDK9. However, if we treat with epoxamycin, we can restore the levels up to approximately 60% of what they were. Yeah. So clearly, what we're doing here is that we are partially, reasonably blocking the proteasome in terms of its ability to be able to degrade CDK9 with the addition of the epoxamycin. And one assumes if you were to go at a higher concentration, we may well be able to increase that. However, you have to also bear in mind that epoxamycin is also pretty toxic, and so it's a balance between getting a readout and killing your cells. So this is similar sort of layout of my slide with regard to CDK6, using the CDK6 PROTAC, again in the MB411 cells, and again treated for 4 hours with both the PROTAC and the epoxamycin. Yeah, in this case, actually that's a typo, but in this case, we're using 300 nanomolar of the PROTAC again on CDK6, and we're basically, by using that, we're basically getting about a 70% knockdown of our CDK6. And then what you can see is that treatment with 1 micromolar epoxamycin restores CDK6 to its full levels. Yeah. Now, two things I wanted to point out here are the fact that in comparison with CDK9, obviously in this case we can restore, or we can prevent the degradation of CDK6 completely with 1 micromolar epoxamycin. But then in addition to that, if you look at the blot plot, which is on the right-hand side, what I wanted to say is that each one of those lanes represents a separate biological experiment. Okay, so I haven't cheated and taken the same cell lysase and put it on 3 times in 3 separate lanes. That's actually 3 separate experiments and 3 separate lysates that have been put, one in each of the lanes. Yeah, so what you can see from that is that our biological reproducibility is really nice. Yeah, and so consequently on the graph on the right, on the left-hand side, excuse me, what you can also see is that we're plotting the mean of the data plus and minus the standard deviation in order to show you that we get really nice tight data. Actually, the same also applies to this graph, the one that you've seen, or to this blot, the one that you saw on the slide before. These are 3 separate biological experiments. So you can see the quality of reproducibility that we can get from our Western blot data on the West, and in this case on the JES instrument. If we think about blocking protein production, then with regard to the cyclohexamide, so in this case again CDK9 PROTAC with MB411s, and we treated them for 4 hours with cyclohexamide, yeah, and we treated them at 10 micrograms per mil of cyclohexamide, yeah, and what we can demonstrate is that by adding cyclohexamide, we can effectively make the PROTAC appear to be more potent. So on that basis, we get DC50 shifting from 11 nanomolar to a DC50 that becomes 1.5 nanomolar. And actually, I think what we're doing here is that we're preventing, or we are reducing the amount of new CDK9 that kinase that is manufactured by the cell in the 4 hours of treatment, and so you can see a more profound effect of the PROTAC. In this case, in this slide, we're looking at CDK6 this time in a similar experiment on the same cells, 4 hours of treatment with cyclohexamide, but this time with the CDK6 PROTAC. But you know what you can see here is that we really haven't changed the DC50 of this PROTAC, and I guess that was sort of reflected in the data that we saw in a couple of slides previously, where actually the CDK6 compound was able to completely wipe out the amount of CDK6 in the cell, whereas we still had some CDK9 remaining. But then again, you know, what I want to point out is that, you know, this is Western blot data. These are graphs that are drawn with Western blot data, and so I think this is really a cool technology and really is applicable to hits to lead screening work, and more importantly, I think, you know, the utilization of this technology not only with cell lines but also with primary cells would be really fascinating from the point of view of, let's say, where you've got blood-borne diseases, and you can therefore isolate PBMCs or T cells from the blood and be able to treat these in well plates with compounds, and then actually to look at these changes that occur to these proteins in those cell types, which would give you data that would allow you to think about future studies in terms of post preclinical phase 1, phase 2. Yeah, so just coming on to issues and problems that are associated and experimental considerations. So the first thing is choose a cell line. And obviously you want to choose a cell line that expresses the appropriate ligase, because there are different warheads, there are different parts of the degrader that will bind different ligases. And so if you're choosing Ceneblom or VHL or one of the others, you want to think about the most appropriate warhead for that. And also in terms of the efficiency with regard to the degradation and the proteasome, so the efficiency with which the compound interacts and the system interacts within that cell. So it could be that it's just a relatively slow turnover or relatively quick turnover, depending on the biological activity of that particular cell. Obviously, some cell types are more susceptible to PROTACs. One of the things to consider is PROTAC permeability. So these are molecules, as I mentioned earlier on, that range from sort of 700 to 1,000 molecular weight and more. So they may actually, their passage into a cell may be inhibited by the chemistry of the PROTAC. And also, one has to really consider that in terms of medicinal chemistry, most chemists drive their pay, if you like, and their bonus based on the potency of the compounds, and really don't consider the efficiency of the compound to remain with the target, so the residency time. And haven't really seen any data as yet to think about whether you need a PROTAC that binds to the ligase and binds to the target and remains there for like 5 seconds, 5 minutes, 5 hours in order to do its job. Also, you need to choose a cell line that really doesn't manufacture your target in mass quantities, because if it does, you're going to find it really difficult to be able to observe a knockdown. So we kind of think about like cell types being in the Goldilocks zone. So they really want to have a little, not too much, not too little, just right when it comes to the amount of protein that they manufacture, which you're trying to deplete. Also, you can't bear in mind the fact that PROTACs can be toxic in themselves, and by that what I mean is that we've had studies in the past where people have gone for proteins that are targets at the top of a biological cascade, and so by removing those proteins, actually then what happens is that the cell becomes pretty dead pretty quickly, because obviously you're removing something which is very essential because it's at the top of a biological cascade. Also, what we tend to know that certain PROTACs have worked because they have very low BCA protein content. And also, if you look down the microscope, you'll excuse my French, but they look pretty crap down the microscope in terms of their appearance. And then you have to really assess different PROTAC incubation times and concentrations in order to minimize that toxicity, because there's nothing wrong with trying to use a PROTAC that kills a cell. If you were, say, looking at a cancer target in oncology, etc., but you'd have to come up with some means of being selective and only killing that cell in comparison with, obviously, the ones that you don't want to kill. So last but not least, loading controls. So loading controls are really important. And I've shown most of the data today has been shown with actin, but we have done different loading controls. You can sometimes see that the toxicity which is created by the effects that you're having on the cell with the PROTAC can result in toxicity that results in a change in your loading control, which isn't really what you want. because if you're trying to ratio to your loading control is changing, then you're going to have issues. And also, cell treatments like epoxamycin can have effects on your loading controls. So what we're just showing here is actually a near-infrared plot of in these MV411 cells when we treated with epoxamycin. And so what we get in the absence of epoxamycin is an amount of actin that gives us somewhere in the region of about 10,000 near-infrared units. However, if we add 1 micromole epoxamycin, then we can deplete that by 40%. So the levels go down to 6,000. So bear that in mind if you're trying to use that as your ratio to that, because obviously that will affect your data and your target protein and those measurements. Second thing is, as I said, that PROTACs can cause toxicity. So in this case, what you can see is that you can see a 4-hour treatment with the CDK9 PROTAC on the left-hand side after 4 hours, either at 100 or 300 nanomolar. Doesn't show any variability or variation in the actin. However, in comparison, on the right-hand side, in those 3 lanes, which are after 24 hours, you can see DMSO looks the same, but the 100 or 300 nanomolar CDK9 PROTAC actually depletes the actin. Yeah, and so that is probably a consequence of the effect that the PROTAC is having in general on your cell population. Next thing is really the different targets that we've looked at, target protein and loading controls. So we've looked at HSP50, we've looked at tubulin, we've looked at vinculin, etc. And so really you need to, I guess, test your PROTAC against these different loading control proteins in order to make sure that they don't have a significant effect before using them to ratio your data to. Again, we had, as I said in the beginning of my presentation, we had Wes for about 4 or 5 years, but what we struggled with was to multiplex a chemiluminescent readout for our loading protein with a chemiluminescent readout for our target protein. And there's 2 reasons for that. One was because, Sod's law says that the molecular weight of your target protein is going to be the same or thereabouts to your loading control, so they're going to superimpose on one another. Or secondarily, that when you come to do it, your loading control is so high in terms of protein concentration that it completely utilizes all of your luminol and peroxide substrates, and so doesn't interact with your target, and you just end up with a blob. And so if you then try and deplete or decrease the amount of protein that you add to your WES, you can see a decrease in, let's say, your actin, but you will completely remove your target protein because you've had to dilute it, let's say, tenfold in order to get a decent blot. And so we actually then moved, as a consequence of that, to JES, because that gave us the opportunity to run multiplex assays where we basically utilize both chemiluminescence generally for our target protein and fluorescence generally for our loading control. Okay, Carsten had just mentioned this, and I just wanted to mention this as well. You can do protein normalization. So left-hand side, chemiluminescence, same blot. The middle of the slide is near-infrared fluorescence, and then the far right is protein normalization. And so that does give you the opportunity then to actually be able to normalize the total amount of protein. The software will do that, the total amount of protein that you put in each lane, and then you could utilize that as well if you wanted to. However, we run BCA assays on each sample, and we don't feel like we really need to do that. Also, I mentioned cell backgrounds. You have to bear that in mind and use a therapeutically relevant cell line. So what I'm showing here on the left-hand side is data that was generated in one cell type or one cell line, and on the right-hand side, data that was generated in another cell type or cell line. And you can see that that the compound with the one compound in triplicate with the three arrows pointing at it is actually the same compound, which shows activity at that concentration in that particular cell line. But the same compound on the left-hand side shows no activity in the other cell line. So you have to bear in mind the cells that you're looking at. Next, you have to also think about the target location, so where your protein is actually exists in the cell, and whether it's in the membrane, it's in the nucleus, or whether it's in the cytoplasm. And that will influence the way you disrupt the cells in order to get the protein out. So sometimes we use Ripper buffer, sometimes we use MP40 lysis buffer, and sometimes we use tris-triton. It really depends on where the target is, the target protein, and the extraction process in order to extract it in order to measure it. And you can see on the right-hand side, we're comparing a target in Ripper buffer, a Ripper lysis buffer versus MP40 lysis buffer. And you can see with regard to MP40 lysis buffer, you hardly see any target, whereas you see a lot of target with regard to the utilization of Ripper buffer, probably because that target is actually in the nucleus. Last but not least, think about the hook effect that can occur in PROTAC. So by that, what I mean is I'm showing a little, not animation, but a little graph on the left-hand side. So really what you want is you want sort of the right stoichiometry between your compound and your target. So, you know, a little bit of compound with a lot of target means that you're going to get degradation, and you're going to get the formation of complexes, appropriate sort of complexes, binary complexes between your target and your compound. If you add too much compound, then what tends to happen is that you end up with, rather than ending up with tertiary complexes, you probably end up with quaternary complexes, where some of the compound is bound to the target and some of the compound is bound to the ligase, and because they're occupying those sites, they actually can't come together and cause close proximity. And so what we showed on the right-hand side there is where we've seen that, where as you increase the amount of compound, it appears as though you see more target in comparison to the control, and that's basically this hook effect. Last but not least, I wouldn't be the managing director of Aurelia Bioscience if I didn't give us a plug. So Aurelia Bioscience are a small contract research organization. We're small, but we're beautifully formed. We live in Nottingham, which is in the center of the UK. We have a number of different biological technologies. We're not a chemistry company, we do biology. And we have another number of different biological technologies within our laboratories in order to design bespoke, tailored assay formats, both cell and biochemical based, in order to do our work. We have access to a phlebotomist, which means that we have access to blood on site, have access to an animal house so we can do ex vivo studies. And we've been in business now for the last 9 years. I originally worked at AstraZeneca, prior to that GSK, prior to that Hospital Roche. So so we've been around the block more than once. So actually that's me finished. And I'd like to say a big thank you to my colleague who's been sitting next to me and prodding me along, which is my colleague Dr. Rachel Deutsch, who I call the Queen of Western Blotting. Rachel did a lot of Western blotting in her previous job using the old equipment that Carsten mentioned. When she joined Aurelia, we introduced her to Wes and Jess. She has 2 children, and the instrument has now become a 3rd child. So she really sees the value in the instrument and technology. And I'd like to thank Hannah from Tokis for organising the webinar, together with the people who we utilise in the UK the most, which is Peru, who's the account manager, and Elaine, who is actually the field application scientist in the UK. So the details there, that's me. If you want to have a look at our website or get in touch, please do so. Thank you for listening, and I'll now hand you back to the Labroots. I'd like to thank them for organising. Thank you.
Thank you very much, Gary, for the great presentation. We will now go on to the live Q&A portion of the webinar. We have just a few minutes left. If you have a question that you'd like to ask, please feel free to do so now. You can just click on the Ask the Question box located on the far left of your screen, and we'll answer live as many questions as we have time for. And then following the webinar, any questions that we haven't answered, we will answer via email with the contact details that you've provided. So to kick off, there's a question here which actually I had the same question. So we'll start with that. So this is perhaps one for Gary. On slide 23, there was an increase shown in the CDK9 levels after treatment with the lower concentrations of the PROTAC compared to DMSO control. And I think there was a comment that the effect was due to ternary complex formation. It was just an interesting effect. It'd be good to get your comments on whether that's a real effect, and whether you have any comments on, yes, that's the correct slide. So yeah, whether you have any comments on what that might be due to.
Yeah, no, thanks, Hannah, you've stumped us.
Sorry, guys.
No, it's all right, it's all right.
I mean, PROTACs do have a tendency to do this sometimes, where there's almost not enough PROTAC potentially. It's binding in low concentrations, and it's almost blocking the protease inhibitor, the proteases, but not. I don't know. We're not quite sure, are we, with this one? Yeah.
Yeah, I think if you mean this, can you see my pointer? Yeah. Probably not. Anyway. If you mean the fact that it seems to go higher than sort of the DMSO control, all we can think is that the PROTAC binding to the protein causes some sort of stabilization event, and thereby we almost like at low concentrations end up with more protein, because the protein has bound the PROTAC and is almost like stabilized, because we've seen it before with other targets, not only CDK9 and CDK6, but other things as well. And it's not an effect which is a consequence of the actin or the local control.
It also might be that we've bound the CDK9 and it's become inactive because this big molecule is now stuck to it, and so the cells notice that, and it's increased its expression of it to try and compensate for the fact that the protein is no longer active, even though it's still present.
Thing is that I always think, the cells don't make proteins unless there's a reason to do it, and also a protein will tend to feed back on itself once it's done its job, or some other mechanism will feed back on itself in order to inhibit, if you like, the manufacture of more protein. And so sometimes I think what we're seeing there is possibly a sort of a disruption of the feedback loop. Yeah.
Because we know CDK9, I think, is quite rapidly expressed. Because when we treated with cyclohex, we shifted the DC50 quite a bit.
Yeah.
So it must be quite quickly turned over.
Yeah.
So if we have disrupted the feedback loop, we might be able to see that at those low concentrations.
Yeah, thank you both. Yeah, it'd be interesting maybe to compare actually to the CDK9 inhibitor alone and look at that effect. But yeah, thank you. That's an interesting point. We are running out of time, and so I think we just have time for one other question. And we have this one in, and perhaps this is one maybe more for Carsten to answer. And the question is, when there is interference among the antibody for the housekeeping protein and the target protein, is it acceptable if the housekeeping protein is run in the same sample in a different capillary?
Hello, yeah, this is indeed acceptable, because in comparison to Western blot, the instrument itself always takes this kind of 50 nanoliters out of each well of your sample. And even if you would have not proper calibrated pipettes, meaning you would pipette, let's say 3 microliters, 4 microliters, 5 microliters from your sample in wells next to each other, you would get the same result. So on our technology, it is, let's say, possible to load your sample in different wells and compare them amongst each other. And in the same way, there was a similar question according like, why don't you use HRP coupled secondaries for target and loading control, which goes in a similar direction? Quite often, your loading control has a much higher abundancy as your target. And even if you got a huge linear range, it might be that your loading control is way too much in those samples. And then you do it the way Gary nicely showed. You swap over for your loading control in the near infrared channel, which is a little bit less sensitive, but by doing so, you have the possibility to detect both together in the same capillary.
Yeah,
Great, thanks, Carsten.
Yeah, I think we would second that. I mean, you know, when we're doing a PROTAC, we typically would use 6 well plates and maybe put between 500,000 and 1 million cells in the well. And then, you know, we tend to cell lyse that in maybe 100 microliters of our cell lysis. And then, but you only really utilize at best like 3 microliters of that in each lane. But then you might have to dilute your protein up to 10-fold in order to do that. So
the thing with the chemi signal, so it's like CDK9, I'll probably need to load 1 mg per ml, 0.5 mg per ml protein to pick that up nicely in chemi. But if I tried to detect actin at that protein concentration, I'd probably get 1 million chemi counts and it would be off the scale. So that's where we end up having to switch to the fluorescence channel for the loading controls, because it was just, you couldn't get the chemi signal for the target and loading control in the right range for that protein.
Okay, great. Thank you both for that answer. I'm aware we are coming out of time, so I'd like to thank the speakers today for their time and the interesting research that was presented. I'd also like to thank Labroots for hosting the webinar today, and also to the audience for joining us and for your attention and interesting questions. There are a number of questions we didn't have time to answer, and those will be answered post-webinar via email. Labroots will be alerting you via email when this webinar is available for replay, and we encourage you to share that email with your colleagues who might have missed today's live event. And with that, thank you all for listening, and until next time, goodbye.